Yeah, so we know that single targeted CD19 CAR T cells are incredibly effective for the treatment of B-cell malignancies, but one escape mechanism is loss of CD19 and development of a clonal escape in a population that is CD19 negative. For many years now, our group has been working with a dual targeted bispecific anti-CD20, anti-CD19 CAR T cells. So this is a follow-up trial where we basically optimize the manufacturing characteristics, increase the depth of lymphodepletion, to basically try to improve outcomes from a prior study. So in the study, we used different reagents to manufacture the CAR T cells, and then we studied varying lengths of manufacturing time. This product was very effective in our clinical trial, which we'll be discussing at our presentation today. The overall response rate was above 90% for all patients, and many of those responses were durable. In terms of toxicity, there was only one grade 3 CRS in the entire population of 29 patients, and only 3 patients with grade 3 or higher neurotoxicity. Some of the exciting findings that we had is that we did find that a shorter manufacturing product did skew the CAR T cells towards a more immature or T stem cell memory phenotype. So we do think that's a better CAR T cell product. And another exciting finding is we had a small phase 2 arm in Mantle cell. We've treated 10 patients, and the overall response rate was 100% in that population. And with a median follow-up of 14 months, not a single patient has relapsed. So really some exciting findings with this dual targeted CAR. We're working with Milwaukee, which is an industrial company, pharmaceutical company, and they're launching they have an ongoing phase 2 multi-center trial to evaluate this product at a larger scale.